Bismillahirrahmanirrahim. Hello everyone. Today we will discuss the protein synthesis. First, we all know that if we extract a cell from the body and then we extract the nucleus, we will find the whole genetic materials is in the nucleus in the form of chromosomes. Okay, the genetic materials in our nucleus is in the form of DNA and other RNA, and we will know this later. Okay, another picture we see here the nucleus that have the genetic materials, the chromosomes, and the DNA. The DNA in its nature is double strand. It's like the ladder that has two backbone and steps in between these backbones. So as we see here, one strand and the second strand. This DNA shape is called double-stranded DNA. If we want to start the process of the protein synthesis, the DNA must not be in this double-stranded shape. It must be unwound. It must be unwound. So we use this, which is called the helicase, that unwinds the double strand into two single strands. Unwind the double-stranded DNA into two strands. Each one is a single-stranded DNA. This is the first step in the protein synthesis. You must know this information that protein synthesis started with break down the hydrogen bonds between the nucleotides. So the DNA double-stranded is unwound and we will start with the single-stranded DNA. This single-strand DNA that we see here, this single-stranded DNA we see here, is formed of nucleotide. GC, GCT, CAT, GAC. This is nucleotide. Each three nucleotide form a gene. This 3-1 is gene number 1. This 3 nucleotide is gene number 2. This 3 nucleotide is gene number 3, and so on. The DNA single strand is formed of nucleotide sequence. Each 3 is a gene. What is the function of a gene? It codes specific amino acid. The gene number 1, code amino acid number 1. Gene number 2, code amino acid number 2. So if you arrange the genes in a specific sequence, you will have amino acid in a specific sequence which will form the protein. So what is the protein? What is a protein? The protein is a long chain of amino acid sequence. The simple structure or the primary structure of any protein is a long chain of specific amino acid sequences. So in this process, each gene codes for specific amino acids. So in a certain specific sequence of amino acids, you will have a protein. So let's read these sentences. This sentence says, the gene is formed of 3 specific nucleotides, as we show here. The specific sequence of amino acids will give a specific protein. Why? Because the protein is formed of long chain of amino acids. Each amino acid is coded by certain protein, which is 3 nucleotides. All right. Here in this slide, we have 3 very important information before we start the game of protein synthesis. First fact is protein synthesis occur in the cytoplasm. Protein synthesis occurs in the cytoplasm. Why? Because all the players that in the game of the protein synthesis are found in the cytoplasm. Okay, the DNA material which codes the protein, which codes your whole characteristic appearance. We know that the whole characteristic appearance, your eyes color, your hair color, your tall, your skin tone, all of this is the your characteristic appearance and the your whole genetic codes is in the DNA. So this DNA is very precious. So it must not leave the nucleus. It must not leave the nucleus. It is very precious source of information. It must be kept in the nucleus. It must not leave the nucleus. But the protein synthesis happens in the cytoplasm. Can we get the DNA out of the nucleus into the cytoplasm? The answer is no. The DNA is kept in the nucleus and the protein synthesis is kept in the cytoplasm. So what can we do? Simply, we produce or make a copy, a complementary copy of DNA is made in the nucleus and then taken out to the cytoplasm for protein synthesis. This picture, we saw here that a certain part of the DNA double-stranded helix is unwound, is opened. Why? Because certain gene we need, a certain gene we needed to be translated or to be encoded into a certain protein. Let's say this human needs collagen. This human needs collagen for hair or his nails and hair. He needs collagen for nails and hair. So we search the DNA for the specific area that have the specific gene for collagen. And then we open the double-stranded, we unwind the double-stranded DNA and expose the gene that is specific for the collagen. And we make a copy of this gene and we take this copy out of the nucleus into the cytoplasm to be translated and produce the protein which we needed here in this example, which is collagen in the cytoplasm. So what we have in here, the first step in the protein synthesis process is called transcription. The transcript is the we call in Arabic it's a nas transcript. يعني GTAC is a transcript. Okay. So transcription is transcript or produce a copy of a section of DNA, a certain section of DNA that we need. We produce a complementary copy of this specific gene. This specific copy of this specific DNA section, we called this we called this complementary copy, we called this messenger RNA. The complementary copy is called messenger RNA. So what we see here is the double-stranded DNA we see here in the nucleus. The pink part of this picture is the nucleus. The yellow part is the cytoplasm. So the DNA, we said that it is kept in the nucleus. So this DNA double helix, we want to copy a certain gene. So this molecule, which is called DNA polymerase, is the responsible molecule to open the DNA molecule. So it opens it into two single-stranded DNA. Okay? Okay. When this single-stranded DNA is exposed, free RNA nucleotide are present in the nucleus. So it came and stuck into this exposed area of the DNA. We call this exposed area of the DNA, we called it template DNA, as a part of the printer process, the printing process. We called it template DNA. So these free nucleotides come here and stick to its complementary bases in the template DNA. And then there is a sugar backbone come and join them together. So the final results will be single-stranded messenger RNA, This one. This is a single-stranded messenger RNA that is formed of nucleotide that is complementary to the template DNA, the template that exposed the area of the DNA that wanted to be copied. Each 3 nucleotide in the messenger RNA is called codon. Do you remember each 3 nucleotides in the DNA was called what? Was called a gene. Okay? But the 3 successive nucleotides in the messenger RNA are called a codon. Okay? Is that clear? This single-stranded DNA leaves the nucleus from the nuclear pore in the nuclear envelope. This is the nuclear envelope, and it has a pore. The single-stranded RNA leaves the nucleus into the cytoplasm to start the game of protein synthesis. So let's revise what transcription is. Transcription is part of the DNA molecule unwind when the hydrogen bond between the complementary base pairs break. This exposes the template strand of the gene that codes for the specific protein being synthesized. Free messenger RNA nucleotide that are present in the nucleus bind to its complementary nucleotide in the template strand. The messenger RNA nucleotide are joined together, forming a single-stranded messenger RNA. Then this copy of the messenger RNA molecule leaves the nucleus. through the pore in the nuclear envelope. You must know that the new strand of messenger RNA is complementary copy of the DNA code from the original gene. So now we will recognize the second player in the game, which is called the ribosome. So the first player in our game is the messenger RNA, the single-stranded messenger RNA. This is the first player. The second player in our game is the ribosome. The ribosome is a structure or molecule found in the cytoplasm. It is very simple. It has a large subunit and small subunit, and then the messenger RNA pass through these two units. This is the second player, which called the ribosome. The third player is called the transfer RNA. The transfer RNA. The transfer RNA is a specific genetic material that is found in the cytoplasm of the cell. It always, and I mean always, carry a specific amino acid. So if we call this transfer RNA number 1, it carries always specific amino acid number 1. Transfer RNA number 1 carry amino acid number 1. Transfer RNA number 2 carry amino acid number 2. Transfer RNA number 3 carry amino acid number 3, and so on. It has another part, which is the anticodon. Do you remember the word codon? It is the three successive nucleotide in the messenger RNA, the copy of the DNA, the complementary copy of the DNA, which leaves the nucleus carrying the same genetic code of the original DNA. We call it the messenger RNA. Each successive nucleotide in the messenger RNA are called codon. Do you remember that? Yes. So the transfer RNA, which carries specific amino acid, it has a specific area which is called the anticodon. The anticodon is complementary to the codon. The anticodon in the transfer RNA is a complementary to the codon. The anticodon in the transfer RNA is complementary to the codon in the messenger RNA. So now you have the three player. So let's start the game. We will combine the transfer RNA, this one here, and the ribosome, which has the large subunit and the small subunit, and the messenger RNA. Again, transfer RNA is the third player, ribosome is the second player, and the single-stranded messenger RNA is the first player. These three players will join together in the cytoplasm to start the game of protein synthesis. All right, okay, let's start. Let's start with the messenger RNA. The messenger RNA strand pass through the ribosome. Between the large unit and the small unit, it passes through it. Or the ribosome moves along the strand. The ribosome moves along the strand of messenger RNA. These are the two players that we know, the ribosome and the messenger RNA. The third player will enter, which is the transfer RNA, carrying its specific amino acid. It leaves or it drop its amino acid. It drop its amino acid in the large unit of the ribosome and leaves the ribosome. Okay, so in this game, the ribosome moves along the strand of the messenger RNA. During this process, each transfer RNA come to the ribosome, carrying its amino acid, drop it in the large unit of the ribosome, and leaves out the ribosome. So what will happen here is the ribosome moves along. The ribosome will move along the messenger RNA in the direction of 5' to 3' direction, because the DNA has a direction. It has two ends. One end is called 5' and the other end is called 3'. This is according to the sugar phosphate backbone that is holding the nucleotide. So let's read this. The ribosome moves along the messenger RNA, reading the next code. So here we have 4. Here we started having what? Having 4 amino acids. This amino acid that was dropped by the transfer RNA. Each transfer RNA come, read the codon by its anticodon, and leave the specific amino acid that it carried. So we move here, the ribosome move along the messenger RNA, the transfer RNA comes and leave its specific amino acid that it carries, and we move along the messenger RNA to read the whole code. Then we will have this long strand of amino acids found in the ribosome. So the result is producing the long chain of amino acids. And this is the protein. The protein is formed of a long chain of amino acids. This is how the game of the protein synthesis happens in the cell. Thank you. See you in the next video.